Matthew Michelson and Franklin Stell in 1958 used ammonium chloride as a source of nitrogen and cesium chloride to prove that replication of DNA is semi-conservative. When a solution of cesium chloride, which is very dense, centrifuged at a high speed, it forms certain bands on the basis of density. The bands which live at the top of the solution are light bands having low densities, and the bands which are formed at the bottom are of heavier densities. So density increases from the top to the bottom of the solution. So these are the bands which are formed when a cesium chloride solution is centrifuged at a high speed. Now, if you mix up DNAs of different densities with cesium chloride and centrifuged together at the high speed, then DNA containing nitrogen 14, that means light DNA, will settle at the top and the heavier DNAs will settle at the bottom. The light DNA will be at the top of the solution. The heavy DNA molecules will settle, they sediment at the bottom of the solution. So two different DNAs are separated along with cesium chloride on the basis of their densities. Lighter DNA, heavier DNA, they can be easily separated by this technique, and this technique is called cesium chloride density gradient centrifugation technique. When centrifuged at high speed, the DNAs are separated on the basis of their densities along with cesium chloride. The scientists were able to create light DNA and heavy DNA separately for their experiment.